appreciate the opportunity to be here. I feel a little like a fish out of water in that I hadn't really thought of what we do as being part of micro and nanotechnology, but I guess since we work with microbes, I guess that is microtechnology in a way. I want to speak to you today about technology that we've developed where we actually, there's a lot of biomolecules that we're interested in within bacteria and or the bacteria themselves, and we're working with livestock in the beef and dairy industry, where it's a very harsh environment, and we're actually using our technology to help the bacteria protect themselves and survive in that environment. Sage, just a brief bit about us. We're an Alberta-based private company. We're headquartered here in Edmonton. Our focus is on developing products for the improving the nutrition and health of food-based animal agriculture. We do not go off onto the pet side as of yet. We're sticking within our where our knowledge base is, and we have stuck with primarily with the dairy and beef industry. I'm going to have to just go briefly through a real quick tutorial for those of you who don't understand what goes on in the rumen of a cow. We're basically dealing with the complex ecosystem where we have three major groups. We have the bacteria, the fungi, and the protozoa, and Sage works directly or indirectly with all three of those groups in the type of technologies that we're developing. Bacteria are the most diverse and numerous group within the rumen, and they're the primary digesters of the complex carbohydrates that allow the animals to consume the type of feeds that they do consume. What most people don't recognize is that they're also the major contributor to the protein supply of the animal. The bacteria produce protein, their own protein. It flows to the small intestine where it's subsequently digested. So they're a huge contributor to the protein supply of the animal. The protozoa represent a huge chunk of the biomass. Because they're larger, they don't represent as much in terms of a culture count basis, but they're a huge contributor to the biomass. And there are really two groups. There's what I'll term the herbivores, which are involved in the digesting of the complex carbohydrates, and the ones that we have trouble with are the carnivores, and they're pretty indiscriminate consumers of the bacteria and just about anything else that is of the right particle size. If anyone comes away with this with a great idea as to how to get rid of these carnivores, I'd be happy to talk to them, because that would be worth millions of dollars a day in North America to be able to control those. The fungi are not so numerous, but they are probably the most potent source of the most potent fibrolytic enzymes that have been evaluated to date. So if it's not necessarily a good production system for producing fibrolytic enzymes, but certainly a great genomic base to go looking for good candidates. Bacterial breakdown in the rumen. About 20% of the bacteria are broken down on a daily basis within the rumen, and that may not seem like very much, but that consumes a great quantity of both energy and is a loss of protein that is lost to the animal, and subsequently we have to feed that in terms of other feeds that we wouldn't otherwise need to use. About over 90% of that breakdown is due to the protozoa. So our activities have been basically trying to work around that protozoal breakdown to enhance the bacterial survival. And the protozoa use lysozyme as their main source of breaking those bacteria down. And when we have a natural system, the bacteria are always exposed to the stimuli of all the various other organisms and the media that's around them. they're constantly producing their whatever defense mechanisms they need to circumvent the things that are trying to outcompete them in that environment. However, when we grow organisms in pure culture, such as when we're producing using a large-scale fermenter to produce, say, a probiotic organism, that organism is not exposed to any of those external influences, and therefore they don't produce those defense mechanisms. Because lysozyme is a necessary component to protozoal breakdown, our technology involves using lysozyme to generate that bacterial defense mechanism in organisms that are grown in a culture-based system. So what we do is our technology is we take, basically it works with just about any gram-positive organism. We grow it in the presence of very low levels of lysozyme. That stimulates the bacteria to produce a stronger cell wall, stronger encapsulation, and significantly reduces the rate that those bacteria are broken down. We've gone from degradation rates as high as 20% per hour down to less than 5% per hour. And that 5% is a very important number, because if a bacteria is broken down faster than 5%, it will not sustain itself in the rumen environment. So we have to drop below that 5% in order to keep an organism in the system. Okay. So the first application that we looked at was for rumen-protected lysine. Lysine is the first limiting amino acid in milk production and beef production. We can't just go and feed extra lysine as we do with, in the case of chickens and pigs, where we just feed more lysine in the diet. Lysine is broken down by those bacteria very rapidly, so we have to come up with a way of protecting it to get it through the rumen. And coming up with a rumen-protected lysine has been kind of the holy grail for dairy cow nutrition for 20 going on 30 years now. This just shows the improvement we get in milk protein yield when we can provide that lysine to the animal. That improvement is, in North America, is worth somewhere between $2 and $3 million a day to the end user in the dairy industry. Lysine production commercially, it's done with Corynebacteria glutamicum. It is arguably the most important industrial bacteria out there. It's used to produce everything from MSG, lysine, just about any other amino acid that is industrially available comes from this organism. Very simple soil organism, but it's been mutated to produce a wide range of products. Normally, the strains that are used for lysine production have an exporter gene that has them excrete that lysine out into the medium. They're harvested, the lysine is extracted from the medium, and then we get the purified lysine. In our case, we have a strain that's deficient in that exporter gene, so it accumulates the lysine within the cell. So we basically generate a bag full of lysine. Okay? But now to get that, we have to get that through the rumen, so we grow that in the presence of lysozyme, and then now that it's hardened, and then we can drive that product through the rumen so that the animal can absorb that lysine and obtain the value that we're trying to deliver. Just as an example, this organism is the original type strain that was isolated back in the 1920s. This is the organism we're working with. So this organism doesn't really produce lysine, but it's the original strain. Growing it in the presence of lysozyme or not doesn't really have much of an impact, but it doesn't produce any lysine, so it's not of value to us. This is our organism, the one that incorporates the lysine within the cell. We can go from 4% all the way down to 1.2%. This is a huge increase in ability to get that organism through the rumen. So in our case, we're interested in lysine, but this technology can be applied to any other type of metabolite that we can get within that cell that's important to deliver to the intestine of the animal for absorption. And down here at the bottom, this is a rumen bacteria that we use as our control. Without the lysozyme, it's 6% per hour, but then when we expose it to the growth with the lysozyme, you can see it drops it below that magic 5%. So even the rumen bacteria that we know that exists in the rumen, without the lysozyme present or the natural stimuli, they're going to be unable to survive as well. Another application that we're using this for, and this was a secondary one, but it's actually the one that we have just actually licensed and has gone into commercial production in the last quarter. Probiotics are used extensively for improving animal performance and as a way of pathogen control. There's a lot of concern over E. coli and Salmonella in the beef sector. There's other organisms as well, but those are the ones that grab all the headlines. We can, by having certain organisms there, there's a number of theories as to why they work, but they actually will reduce the shedding of these pathogens so that we can have a safer food industry. One of the unfortunate parts is that the marketing, both on the human and on the animal side, is way ahead of the science, and that's one of our struggles, is to try to, we're trying to be a science-based company, and there's a lot of marketing out there that, well, everyone's familiar with, I'm sure you've heard of Bifidobacteria regularis, which is really a marketing name. There is no such organism by that name, but it sells a lot of product. Responses can be quite variable when we're dealing in animal production, and of course that's the big driver. Whether you feel good or not, that will impact your decisions to buy something in the store. But at the end of the day, the farmer looks at it and says, did they grow faster or not? And so we're dealing with that. And the poor survivability of these probiotics is a big contributor to that variability. But this is the some of the data that led to getting our license. We dealt with this is not really zero down here, but it's down in the single digits for survivability coming out of where we looked at it's hard to culture these organisms individually when they're in a complex milieu like the rumen content. So we looked at the DNA that was survived getting out of the rumen, and very little of the DNA was escaping when you just used the organism by itself. But when we had preconditioned it, grown it in the presence of the lysozyme, hardened up that bacteria, basically encapsulated it so that it protected it from the predation within this rumen system, we were actually getting 3 times as much of the bacteria leaving the fermenter as we were putting in every day. So we actually got the organism to grow, survive, thrive in those conditions, whereas in the case of the control organism, it was surviving, it was getting through, but at a very small level. So that's where we are today. I would like to thank our collaborators and contributors to what I would consider our success to date. A group is only as good as the team that is part of that makes that whole. And we've had a lot of. We started out about 8 years ago, and things didn't really get going until we established our relationship with what was at that time ARC, but is AITF. And a big thanks goes to Janet Marowich and Dr. Shee of AITF. We've also had support from the NSERC IRAP program, Agriculture and Agri-Food Canada in Lethbridge, and Keystone Labs here they're stationed here in Edmonton as well.